Hey guys, and welcome back to SciShy. Now, before I get into this video, I want to let you know that I've compiled all of my past videos, as well as some practice experiments that you can actually try out on your own at scishy.com. Now, let's get back into this video. We're going to be talking about mini-preps, which allow scientists to isolate plasmid DNA from bacteria. And now, let's get into some underlying concepts behind this process. So this is our bacteria. And as you can see here, we have our chromosomal DNA, and we also have these circular pieces of DNA, and these are plasmids. Now, plasmids are extra chromosomal DNA, which are found as circular, double-stranded nucleic acids. We know that almost always, double-stranded nucleic acids point to DNA, and as you can see, they are circular. Now, extra chromosomal helps us understand that they're outside of our chromosome. But what do bacteria use these for? Well, let's take our example of this bacterial population. This singular green bacterial cell is antibiotic resistant, while all of these brown or gold ones are not resistant to antibiotics. Well, if this population were exposed to antibiotics, all of these cells would die, except for the singular green one. But plasmids allow the cell to pass on antibiotic-resistant genes to other cells in its population. And so this population would effectively turn into almost an entirely antibiotic-resistant population. And so when you have antibiotics, these are shielded from antibiotics, while cells without this plasmid, let's say this population, end up dying off. And so it allows actually genetic diversity to occur in bacterial populations. And that's one key characteristic of plasmids. Well, how do scientists use this structure in real life? That is where we get into restriction enzymes. This is our circular plasmid. And as you can see here, we have another gene. Let's say this gene is for insulin, or the creation of insulin. And insulin is commonly deficient in diabetic patients, and so it's important that we can make this protein. So let's say we have our plasmid and insulin. We can use a restriction enzyme, which I'll be going over in another video, to cut our plasmid and insert this gene. And then you can effectively have a plasmid to produce insulin. And we can then insert it into other bacteria and have bacteria produce insulin for us, and allowing us to produce this protein for diabetic patients. Well, mini-preps are key parts of this process because mini-preps allow us to replicate this plasmid rapidly. Now let's go over what a mini-prep process is. First off, we have a bacterial population, and these bacteria are inserted with this plasmid. These cells are then lysed, so these are directly removed from the culture and harvested. After they are centrifuged, the DNA is bound. This allows us to wash away other parts and the DNA to be bound to a silica column for us to extract later on. Then we wash it multiple times, and at the end, they are eluted, which allows plasmid DNA to be ready for transfection. Now we can quantify this using a spectrophotometer, which allows us to see how much plasmid DNA is produced. So let's go over this entire process. First off, we talked about mini-preps and the concepts behind them, as far as what are plasmids, how are plasmids used in bacteria. Then we also talked about restriction enzymes and how they are used in real life, and how mini-preps fit into this, where we can rapidly produce plasmids through this process. Well, this is a really short video, but I want to thank you for listening and hope you tune into the next one. Thank you.